All right. Welcome, everyone. I think it's safe to say you're all here because you really like getting to the bottom of things, right? Especially when it comes to new scientific discoveries.
Absolutely.
That's what we try to do with each of these deep dives.
Exactly.
Today, we're diving into some research that you sent our way.
Yeah.
All about how our cells age, how that puts stress on their, powerhouses, the mitochondria.
Yeah.
And how all of that triggers inflammation throughout the body.
That's right. This paper lays it all out. It looks at how aging at the cellular level, when combined with that stress on the mitochondria, those energy-producing units inside our cells,
Yeah.
it can set off a huge inflammatory response.
Okay, so to put it simply, we're talking about what happens when our cells get older. They enter this state, it's called cellular senescence.
Right.
And then the mitochondria, the energy factories, when they're under stress, they actually start inflammation.
Yeah, and what's really interesting
This is crazy.
it hints at possible new ways we can actually step in and maybe change these processes. Wow. They used a whole range of techniques in this research. High-resolution imaging of human cells, super detailed studies with mice.
Oh, wow.
To really dig into how this all works.
So what we're going to do today is pull out the most important insights, right? Like, how does this mitochondrial stress lead to inflammation as we age?
Yeah.
And what does it all mean for us if we want to live healthier, longer lives?
Exactly.
Okay, so first things first. Cellular senescence.
Right.
What is it, and why should we care?
Well, cellular senescence, basically, it's when a cell just stops dividing permanently. And it's not because it's been damaged to the point of, death.
Okay.
It's more like a programmed stop.
So it's like a built-in, okay, we're done here type of thing.
Exactly. In response to different stresses, like DNA damage or even just wear and tear from, just living.
Over time.
Yeah.
Makes sense.
But the thing is, these senescent cells, they're not just sitting there doing nothing. They're actually quite active in secreting this complex mix of molecules. We call it the senescence-associated secretory phenotype, or SASP for short.
SASP, got it.
Yeah.
So these older cells are basically pumping out a bunch of different substances.
Right, and that's where things can start to go wrong.
Okay, I see.
These secreted factors, they include things like inflammatory cytokines, growth factors, and proteases. And while the SASP can actually be helpful sometimes, like when you're healing a wound,
Oh, interesting.
the issue is when you get a buildup of senescent cells as we age or with chronic diseases.
Yeah.
This constant stream of inflammatory molecules can cause lingering, low-grade inflammation in your tissues.
That's not good.
Not really. It's actually linked to a lot of age-related problems.
Okay, so now we got to bring in the mitochondria. These are the powerhouses of our cells, right?
That's right. They're essential.
So how are they connected to these senescent cells and this whole information thing?
Well, mitochondrial dysfunction, when the mitochondria aren't working right,
Yeah.
that's a big hallmark of cellular senescence. As cells age and experience stress, their mitochondria, they become less efficient.
I can see that happening.
And this decline, it seems to be tightly linked to the development of senescence and even its maintenance.
Interesting.
Some studies have even suggested that if we could target these mitochondria, we might be able to develop therapies to fight the effects of senescent cells.
That makes sense. So just to recap, we have older cells, mitochondria that are getting sluggish, and this inflammation going haywire.
Exactly.
But you mentioned some new research that really focuses on a specific type of stress on the mitochondria.
Right.
Something called MyMMP, right? What is that?
This is where it gets exciting. They found that in senescent cells, there's this special kind of sublethal mitochondrial stress. They call it minority MMP or MyMMP.
Okay, MyMMP.
Now, you may have heard of MMP before. It stands for mitochondrial outer membrane permeabilization.
Oh, right.
And it's a crucial step in apoptosis, which is programmed cell death, like the cell's self-destruct sequence.
Wow.
But my mom is different. It's milder. It doesn't kill the cell outright.
So it's like apoptosis light.
Yeah, kind of.
Not a full shutdown, just some things going wrong in a few mitochondria.
Exactly. In my mom, the outer membrane of only some mitochondria becomes permeable, leaky.
I see.
Unlike full MyMPOM, this doesn't immediately kill the cell. And they found two key proteins are involved here, BX and BEAK.
Okay.
These proteins, they're known to create pores in the mitochondrial membrane during full apoptosis. But in my mom, they seem to form smaller openings.
So it's like a controlled leak.
You could say that, and one of the things leaking out is mitochondrial DNA, or mtDNA.
mtDNA leaking out, and that's a problem.
It can be, yes. Now, in apoptosis, mtDNA release also happens, but the inflammatory effects are usually controlled by caspases, those enzymes that break down the cell during cell death. But in senescent cells going through mimone, this control mechanism isn't as strong. The researchers actually found a lot more mtDNA in the cytosol of these senescent cells, the fluid inside the cell, outside the nucleus.
Okay, but how did they know it was mtDNA specifically, not just any DNA that was floating around in there?
Well, they used a few different techniques. With super-resolution microscopy, they could actually see more DNA-containing structures, called nucleoids, in the cytoplasm of senescent cells.
Wow, that's amazing.
Then they used a technique called qPCR.
Okay.
It measures the amount of specific DNA or RNA.
Right.
And they found higher levels of a particular mtDNA region in the cytosol. Importantly, they also showed that this DNA was usually attached to TFAM.
TFAM.
It stands for transcription factor A, mitochondrial. It's a protein that binds to mtDNA and packages it inside the mitochondria.
So this TFAM being there means the DNA probably came from the mitochondria.
Exactly, that's what they figured. They even found higher levels of TFAM itself in the cytoplasm.
Interesting. So the paper mentions this TM-bound mtDNA is really good at setting off a specific pathway.
Yes, this is a crucial point. They found that this type of mtDNA is a strong activator of the CGAS STING pathway.
Okay, and what is that exactly?
It's basically a surveillance system in our cells. It detects foreign or misplaced DNA and triggers an inflammatory response.
This is like the cell's security system.
Yeah, a good analogy. And this response includes producing those signaling molecules that make up the SASP.
Right, that cocktail of stuff we were talking about.
Exactly.
Okay, so let's put it all together. My MP lets mtDNA escape. It's spotted by CGAS, which alerts tinging, and boom, we have inflammation.
That's a great summary.
But is MyMom really the cause of the SSP, or is it just another thing that happens in senescent cells?
Well, the researchers had some pretty convincing evidence that MyMom is a key driver.
How so?
They used this drug, ABT-737. It's known to induce MyBOMP.
Okay.
They gave it to normal cells, cells that were still dividing. Even though it didn't kill the cells right away, over time, this low dose of ABT-737 caused them to release more inflammatory cytokines, like IL-6 and IL-8.
Uh-oh.
And it even made those cells become senescent faster.
Oh, wow. So it's like a vicious cycle. Triggering MyMomp makes cells pump out inflammatory signals and even pushes them toward senescence.
That's right. Now, remember those BAX and beta proteins? They're really important here.
Yeah, they're the ones making the holes, right?
Exactly. The researchers showed that these proteins are essential for mtDNA release and the SSP.
How did they figure that out?
They used CRISPR-Cas9.
Oh, the gene editing tool.
Yep. They created cells without DAG and bake.
Click.
Even when they triggered senescence in those cells, there was way less mtDNA floating around.
Interesting. So no bags and bake, no leaky DNA. Makes sense.
And when they looked at the genes being expressed and the cytokines being produced, they saw a big reduction in SASP factors when BEX and bake were gone.
But did taking out BAX and bake just stop the cells from becoming senescent altogether?
That's a great question, and they checked for that. Deleting BAX and BAC didn't affect the markers of cell cycle arrest.
Meaning what?
Meaning the cells still became senescent, they just weren't as inflammatory.
Ah, I see. So it's really the leaky mitochondria causing the trouble, not just the fact that the cells aren't dividing anymore.
Exactly. It's a very important distinction.
So it seems like we're really zeroing in on BAC and this whole MyMom thing as the culprits behind senescence-driven inflammation.
That's right. And they looked into other possible causes, too, like the mitochondrial permeability transition poor or MPTP.
Right, another channel in the mitochondrial membrane?
They used a drug that blocks MPTP, but it didn't change much.
So not the MPTP.
Yeah.
What about other things linked to messed up mitochondria?
They considered those, too, like those cytosolic chromatin fragments and reactive oxygen species.
I vaguely remember those from bio class.
But even when those were present, deleting BAX and BAC still reduced the SASP. It was really consistent.
Very interesting. So BAX, BAK, MyMomP, these are the big players.
They are. And what about that CGAS STING pathway? Remember that?
Yeah, the cell's security system that gets triggered by mtDNA.
Well, they found a lot of evidence to support that link, too. They saw CGAS hanging out with that TFAM-bound mtDNA in the cytoplasm.
Like right there at the scene of the crime.
Exactly. And when they got rid of CGAS or STING, the cells produced way less IL-6 and IL-8.
The inflammatory signals.
And when they purposely put mtDNA into normal cells, it caused inflammation, but much less so in cells without STING.
Wow. Okay, so the whole chain of events is pretty clear. Stress leads to MyMop, back X, and back make the holes. mtDNA escapes, CGS spots it, sting gets activated, and then inflammation goes crazy. It's a whole cascade.
It is a really intricate one.
Now, the paper also looks into why MyMop only seems to happen in some mitochondria.
Right. So they started investigating mitochondrial dynamics.
Like how they move around and change shape.
Exactly. It's a constant process of fusion and fission, joining together and splitting apart.
Okay.
And they found that senescent cells tend to have a lot of hyperfused mitochondria.
Meaning?
Meaning they're longer, more connected, less fragmented.
Huh. Interesting.
But then they looked closely, and it turned out those few fragmented round mitochondria, those were the ones without cytochrome C and with lots of activated BAX.
So those are the leaky ones?
Exactly. Which made them think that maybe the long interconnected networks are actually protective.
Makes sense, like they're stronger together.
Right, and the ones that break off, those are the troublemakers.
Okay, so how did they test that idea?
They messed with a protein called MFN2, which is involved in fusion.
Okay.
When they lowered MFN2, they got more fragmented mitochondria. And in those cells, they saw more MyMom, more mtDNA in the cytoplasm, and more inflammation, all without affecting cell cycle arrest.
So blocking fusion made things worse.
Exactly. And the opposite was true, too. When they forced the mitochondria to split apart using a chemical called CCCP, they saw more mtDNA release and more SFP.
Okay, so mitochondrial dynamics, how they fuse and split, that really matters for MyMengP and inflammation.
It really does.
This is all super fascinating, from the scientific point of view.
Right.
But what about real-world stuff? Can we actually do anything with this knowledge?
That's the exciting part. The researchers looked at potential therapies based on all these findings.
Like what?
Well, they focused on BAX since it's so important for MyMomP. They tested a BX inhibitor called BAI1.
So blocking BAX, basically.
Yeah, and it worked. BAI1 stopped mtDNA release, BAX activation, and the SAFP in senescent cells.
Wow, that's impressive.
And it was specific to BAX. It protected cells without B from a certain type of cell death, but it didn't affect inflammation caused by other types of DNA.
Okay, so it was really targeted. And did they try it in actual living organisms?
They did. They gave BAI1 to old mice for 3 months.
And what happened?
Those mice actually showed some amazing improvements. They had better coordination, better grip strength, and they were less frail.
That's fantastic.
And the best part, their lifespan didn't change. They were just healthier for longer.
That's incredible. So healthier aging without necessarily extending life.
Yeah.
That's the goal, right?
Exactly. And they found lower levels of those SAP factors in the treated mice's blood.
So less inflammation overall. And were the benefits limited to muscles?
Nope. Their bones were better, too. BAI1 improved bone density and structure in the older mice.
Really?
And there were fewer inflammatory markers in their bone tissue.
So it's like this BII1 was hitting multiple targets at once.
It seems that way. And get this, they even saw reduced inflammation in the brains of those treated mice.
Wow, that's amazing.
And they confirmed that BAI1 can actually cross the blood-brain barrier.
So it can reach the brain directly. That's huge.
They even did some really fancy analysis of brain cells and found that BAI1 reduced the number of senescent cells in the brain.
So it's not just stopping inflammation, it's actually tackling the source of the problem.
That's what it looks like.
Okay, wow. So inhibiting BAX, blocking this MyMP pathway, it really seems to have a positive impact on a lot of tissues that are affected by aging.
It really does, which makes this research so exciting. It strongly suggests that this mitochondrial stress in senescent cells is a major driver of age-related problems.
All right, so for everyone listening, let's sum up the key takeaways here.
The big discovery is that cellular senescence involves this sublethal mitochondrial stress, it's called MyMomP, which is caused by the proteins BAX and BAKE. MyMomP causes mtDNA to leak into the cell's cytoplasm, where it's seen as a danger signal. That activates the CGA sting pathway, which then leads to the SASP, all those inflammatory molecules. And importantly, this inflammatory aspect of senescence driven by MyMomP seems to be a huge part of why our tissues don't work as well as we age, even more so than the fact that cells stop dividing.
And it's really wild to think that BX and BAY, these proteins that are usually involved in cell death,
right,
they're actually playing this totally different role in senescence where cells are resistant to dying.
I know. It really changes how we think about these two cellular processes.
And the fact that we might be able to use this knowledge to develop therapies, that's huge.
Absolutely. Targeting my mom, like with that BX inhibitor BAI1, it's showing real promise for reducing inflammation and improving health span, and it seems to work in a lot of different tissues.
So, to leave you with something to think about, this research is telling us that aging isn't just about cells losing their ability to divide.
That's much more than that.
It's also about these subtle chronic stress signals coming from inside our cells, from the mitochondria.
Right, and who knows what amazing discoveries and treatments are still to come.
Exactly. It's a really exciting area of research. Thanks for joining us on this deep dive. We'll see you next time.